Okay, going through the short answer questions. So this first question about the forest. Two variables other than species richness. So the obvious one to mention is species evenness. The other things that the MARC scheme had was looking for the area occupied by the forest or the change in the area occupied by the forest, the range of a species, so where one species is found changes, and the number of threatened species. That was what was on the marking scheme. Then in this question, outline the reason for the loss of a dipterocarp forest, and there's 2 marks there, so you need to be making 2 points. So the first one would be some kind of anthropogenic reason, so human activity, logging, removal of trees, that gets you 1 point. And then the second point is the sort of the why, so to sell timber or to grow crops or for plantations or for agriculture. So that's your second mark. Then this one about peat, there were kind of 2 ways that you could go about doing that. You could either talk about it in terms of there's increased combustion, so that's going to be more carbon dioxide, greater climate change, and that's going to have an impact on the biodiversity. Or you could talk about it in terms of the actual loss of an ecosystem and a habitat. So you could talk, which is what most of you did, which is talking about the fact that by removing the peat, you're removing a habitat. Animals would then need you're removing probably plants within that habitat, and then animals would need to find other places, adaptation, failure to adapt fast enough, extinction. Okay, question 13. With this one, this was a bit of a sink or swim question. Generally, people either got 3 out of 3 or 0 out of 3. And the most common reason for people getting 0 out of 3 was not labelling what they're drawn. I appreciate that the question says draw and not draw and label, but in biology you always label, okay? Because otherwise it's just lines on a page. So the grey line was the cell wall, because the question says within the diagram. So this is the cell wall, okay? You don't get a point for labelling that because it's not an internal structure. So then the things that you could have done, oh goodness, sorry, this is a terrible drawing. So that would be the cell membrane, then that would be the nucleoid region, plasmid, 70S ribosomes. Now, if I just left it like that, that is nil point, okay? But if I do now cell membrane, 1, 70S ribosome, 2, etc. Okay, so you've got to add your labels. Now, electron micrographs can be a bit tricksy because they can be quite blurry and hard to work out what's going on. And I'm afraid that is just what electron micrographs look like. So you've just got to sort of use your deductive reasoning as well as your really, really close observations. So number 1, big organelle, clearly delineated from the cytoplasm, different consistency on the inside. That can only be the nucleus. Okay, so that's really the only option there. Okay, so now if we look at number 2, I'm just going to zoom in, which I know is cheating because obviously you can't do that on the exam, but just to sort of show you what you've got there is you've got flattened membranous sacs, clearly distinct, so they're in a distinct area on their own, and with quite a lot of vesicles around them, and so that makes that the Golgi. Sorry, I don't know what's going on here with my pen, so that makes that the Golgi. Now I've got a different type of pen, that's delightful. And then this one, okay, so again I'm going to just zoom in. Oh no, I'm not apparently. Anyway, but what you've got there is a kind of an oval shaped organelle with some markings on the inside. There's quite a lot of them, so there's 1, 2, 3, 4, and so that indicates it is a mitochondria. Okay. And then this next one says using evidence from the micrograph. Okay, so that means you can only use things that you can actually see in the picture. So you can say that it's eukaryotic because there are membrane bound organelles, because you can see those. That would be 1 point. You can say eukaryotic because the nucleus is present. You can see those. Eukaryotic because a mitochondrion is present. Eukaryotic, you could talk about the size. So the scale here, that tells you that's 400 nanometers. So the whole thing is clearly going to be much larger than that. And so that gives you an indication that it's eukaryotic rather than prokaryotic. And then for C, it is asking you for the function of the flagella and the ribosomes in prokaryotic cells. So the flagella is for movement and the ribosome is for protein synthesis. And that brings you to the end of section B.